Biotechnology harnesses the power of biology for practical applications. It involves manipulating living organisms or their components to create valuable products and technologies. Biotechnology is not new. For millennia, humans have used yeast for bread making and bacteria for fermenting food. What is new is our ability to understand and manipulate the genetic code itself. Modern biotechnology, driven by genetic engineering, has revolutionized various fields. It impacts medicine, agriculture, environmental science, and more. Biotechnology offers solutions to global challenges, such as disease, food security, and climate change. This essay will explore the fundamental principles and processes of biotechnology. We will delve into genetic modification and cellular techniques, providing illustrative examples from medicine and agriculture. We will also examine the profound impact of biotechnology on addressing global issues and discuss the potential advancements that lie ahead. To grasp biotechnology, we must first understand DNA, the blueprint of life. DNA, or deoxyribonucleic acid, is a complex molecule found within the cells of all living organisms. It carries the genetic instructions necessary for an organism's growth, development, functioning, and reproduction. Imagine DNA as a long, twisted ladder. The rungs of this ladder are made up of 4 chemical bases: adenine, thymine, guanine, and cytosine. The specific sequence of these bases determines the genetic code, much like the order of letters determines the meaning of a word. Genes, specific segments of the DNA sequence, hold the instructions for building proteins. Proteins are the workhorses of the cell, carrying out a wide range of functions. They act as enzymes, hormones, structural components, and more. Understanding the structure and function of DNA is fundamental to biotechnology. By manipulating DNA, scientists can alter the genetic makeup of organisms and engineer them for specific purposes. Genetic engineering is a cornerstone of modern biotechnology. It involves the deliberate modification of an organism's genetic makeup to introduce new traits or enhance existing ones. This powerful tool allows scientists to manipulate the very essence of life, tailoring organisms for specific purposes. One widely used technique in genetic engineering is recombinant DNA technology. This involves cutting and pasting DNA segments from different sources to create new combinations. Scientists can insert a desired gene from one organism into the DNA of another, endowing it with a new trait. For example, the human insulin gene can be inserted into bacteria. These genetically modified bacteria can then produce large quantities of human insulin, which is used to treat diabetes. This breakthrough has revolutionized diabetes management, providing a reliable and affordable source of this essential hormone. Genetic engineering finds applications in various fields, from medicine and agriculture to environmental science and industrial biotechnology. It holds immense potential for developing novel solutions to global challenges. Section 4. Cellular Engineering, Manipulating the Building Blocks of Life. While genetic engineering focuses on modifying DNA, cellular engineering takes a broader approach. It involves manipulating cells themselves, modifying their structure, function, or behavior. This field encompasses a wide range of techniques, from cell culture and cell therapy to tissue engineering and bioprinting. Cell culture is a fundamental technique in cellular engineering. It involves growing cells outside of their natural environment in artificial conditions that mimic the body. This allows scientists to study cells in detail, test the effects of drugs, and develop cell-based therapies. Cell therapy involves transplanting living cells into a patient to treat a disease or repair damaged tissue. This approach holds great promise for treating a wide range of conditions, from cancer and autoimmune diseases to spinal cord injuries and organ failure. Tissue engineering takes cell culture a step further, aiming to create functional tissues and organs in the laboratory. This involves seeding cells onto scaffolds, providing them with the nutrients and signals they need to grow and organize into complex structures. Section 5. Biotechnology in Medicine, Revolutionizing Healthcare. Biotechnology has revolutionized medicine, leading to groundbreaking treatments and diagnostic tools. From gene therapy and personalized medicine to the development of novel vaccines and antibiotics, biotechnology is transforming healthcare as we know it. Gene therapy, still in its early stages, holds immense promise for treating genetic diseases. It involves replacing or inactivating faulty genes that cause disease. While challenges remain, gene therapy has the potential to cure diseases that were once considered incurable. Personalized medicine, another exciting frontier, tailors treatment to an individual's genetic makeup. By analyzing a patient's DNA, doctors can predict their risk of developing certain diseases, diagnose diseases earlier, and select the most effective treatments. Biotechnology has also played a crucial role in developing new vaccines and antibiotics. Recombinant DNA technology allows scientists to produce vaccines that are safer and more effective than traditional vaccines. In the face of rising antibiotic resistance, biotechnology is essential for developing new antibiotics to combat deadly infections. Section 6. Biotechnology in Agriculture. Cultivating a Sustainable Future. Biotechnology plays a crucial role in addressing global food security and promoting sustainable agriculture. From genetically modified crops that increase yields and reduce pesticide use to precision farming techniques that optimize resource allocation, biotechnology is transforming the way we produce food. Genetically modified, or GM, crops have been a subject of much debate, but their potential benefits are undeniable. GM crops can be engineered to resist pests, tolerate herbicides, withstand harsh environmental conditions, or enhance their nutritional content. By increasing yields and reducing the need for pesticides and herbicides, GM crops can contribute to a more sustainable and food-secure future. Precision farming, another application of biotechnology in agriculture, involves using sensors, data analytics, and automation to optimize crop management. By monitoring plant health, soil conditions, and weather patterns, farmers can make informed decisions about irrigation, fertilization, and pest control, reducing waste and maximizing yields. Biotechnology also contributes to livestock improvement. Genetic selection and breeding techniques can enhance animal health, productivity, and welfare. For example, scientists are developing disease-resistant chickens and pigs that require fewer antibiotics, reducing the risk of antibiotic resistance spreading to humans.